Hello, good afternoon and good morning, everyone. Thank you for joining our Avantor webinar today. My name is Maria Castaño, and I'm Life Science Product Commercialization Manager for Europe at Avantor. I'm very pleased to welcome you all to this webinar, where you will learn about generating complete reference quality microbial genomes with nanopore-only metagenomic sequencing. I'm glad to introduce our speakers to you, Stephanie Acosta from Oxford Nanopore Technologies and Lei Liu from Aalborg University in Denmark. Stephanie and Lei will discuss about the importance of long reads to obtain complete closed microbial genome assemblies, the latest nanopore sequencing chemistry delivering metagenomic consensus accuracies of Q50, also how researchers at Aalborg University are applying nanopore-only approaches to generate affordable reference quality genome assemblies from complex environmental samples. And you will hear also about open source analysis tools such as Nanophase to streamline the generation of metagenome assembled. Now, before I hand over to our speakers, I would like to provide some important information about how the webinar will progress. After the presentation, there will be a Q&A session. Please write and send your questions in the questions chat you see on your control panel. They will be answered in the Q&A session or directly by email if we would run out of time. This webinar will be recorded, and you will get the link by email after the session, so you and your teams can go through it at your convenience and when you need it. Also, in the control panel, you will see a handout section with some PDFs you can download to have additional information on today's webinar topic. At the end of the webinar, you will also receive a survey, and we would appreciate if you could please fill it. It's very important for us to get your feedback to improve the quality of our webinars. And finally, for additional comments or questions, you can always contact us on webinar@avantorsciences.com or visit our website at bwr.com. So thank you all and enjoy the presentation. Stephanie, please welcome, and you can start.
Thank you, Maria. Recent advances in nanopore sequencing mean it's now possible for any lab to generate reference quality. Thank you, Maria. Recent advances in nanopore sequencing mean it's now possible for any lab to generate reference quality microbial genome assemblies without the requirement for short reads or reference polishing, saving considerable time and cost. In this webinar, Dr. Lei Liu from Aalborg University will share his experience utilizing nanopore-only long-read sequencing to generate complete high-quality genome reconstructions from complex metagenomes. We'll start with a brief introduction to nanopore sequencing, how it works, and the recent advancements we've made to our technology to improve throughput and accuracy. Nanopore sequencing is an end-to-end platform that covers sample preparation, sequencing, and data analysis. We have products that fit into each of these spaces, meaning you have maximum ability to control your experiments and answer your biological questions. The platform is flexible, scalable, and easily accessible for any lab. We have different flow cells that offer various data outputs to accommodate your data needs, and all of our devices use the same software and library preparation kits, allowing for an easy transition between our devices. So here's a little bit of background information on how the technology works. Our flow cells contain a synthetic membrane that's dotted with nanopores, and there's also an ionic current that's passing through. During the library preparation process, a motor protein, which is shown here in purple, is attached to the ends of your fragments. This motor protein has a couple of different purposes. It helps to unwind the double-stranded complex and translocate the molecule through the pore, while also acting as a molecular brake by controlling the translocation speed. DNA passes through the pore at around 400 base pairs per second, and RNA at around 80 base pairs per second. Without the motor enzyme, DNA and RNA would fly right through, and we wouldn't be able to read the resulting signal, so the motor protein slows it down. As the DNA or RNA molecules pass through, the nucleotides create distinct disruptions in the ionic current, which we can see here in the figure on the right. We call this squiggle data, and it's provided to you in a FAST5 file format. And then we use base calling algorithms to base call that signal and provide that information in a FASTQ file. And there are a number of benefits to sequencing this way. With Nanopore, you have the option of preparing sequencing libraries via a PCR or PCR-free manner, allowing a native look at either DNA or even RNA instead of a copy. With PCR-free options, base modification information is preserved and can be directly interpreted without the need for bisulfite conversion. This contributes to our data being information-rich, so you can see structural variants and methylation all within the same dataset. We're also read length agnostic, so whatever length your fragments are, that will equal your read lengths. With short fragment mode now integrated with our Minnow software, we're able to sequence fragments as short as 20 base pairs, which is great for cell-free DNA, circulating tumor DNA, and FFPE samples. And we can sequence all the way up to 4 megabase pairs, which is the current customer read length record. Real-time data collection allows you to only collect the data that you need. You can stop your sequencing run at any time point, flush the flow cells, and reuse them for another sample. We also offer a low cost to entry with our starter packs, so you don't need to purchase the device outright. With our starter packs, you're essentially only paying for the consumables, and we're providing you with the device to use as a loaner, and these packages are a fraction of the true instrument cost. With longer reads, you're able to receive more comprehensive data sets. Nanopore sequencing can deliver everything traditional short-read sequencing technologies can, plus provide new insights that were not previously possible. Short reads are limited to single nucleotide variants, including SNPs and indels, and nanopore sequencing can detect these too, but we can also read through complex structural variants, identify copy number variation, identify base modifications, and also phase mutations. And we can do all of that in one single experiment. Now let's dive a little further into the latest updates we've made. We recently released a new pore type called R10.4.1. The difference between this new pore and our standard R9 pore is that it has 2 reader heads within the lumen, and R9 only has 1. With 2 reader heads, we've increased the sensing region within the pore, which allows us to capture a more distinct signal and better resolve homopolymer regions. This has led to our increased accuracy. We also improved the sequencing adapter in our V14 or Kit14 library prep kits. It's a new high capture adapter, meaning that more fragments will receive an adapter, leading to higher yields because more fragments will be sequenced. These updates help to simplify our chemistry offering. You used to require Kit 9 with R9 pores for high yield and Kit 12 with R10.4 flow cells for higher accuracy. Now, the combination of the new R10.4.1 pore and Kit 14 delivers high yield and high accuracy, providing you with the best of both worlds. You can now achieve Q20 simplex accuracy, Q30 duplex accuracy, which we'll explain in just a moment, high quality homopolymer and indel calling, and high yields without having to sacrifice output for accuracy and vice versa. On existing devices, which only need a software update. So there's no new device purchases required to gain access to our new chemistry. This is all built into the V14 kits. So 2 accuracy types exist, simplex and duplex. Simplex is a single pass of just one of the strands of a double-stranded DNA molecule. Duplex is when both strands of that molecule are sequenced separately but then combined to deliver one higher accuracy base call for the single molecule. The R10.4.1 pore with Kit 14 chemistry gives visibly vast improvements over the old R9 chemistry. And duplex data is the cleanest and most accurate data from nanopore sequencing and is independent of read length. Another unique feature to nanopore sequencing is the ability to perform adaptive sampling. This is a real-time method of software-controlled enrichment. It allows for targeted sequencing to happen in silico at the software level, alleviating the need to enrich for your target upstream of library preparation. And there's no limit to read length or target numbers. You only need a reference file for your target or targets of interest. The library is prepared as if you were doing whole genome sequencing. So no enrichment up front. You don't need to design baits, probes, or any primers. You would then turn on adaptive sampling in our Minnow software, and typically a bed or a FASTA file detailing your target regions will need to be uploaded. Our software will map any molecule that begins to translocate through the pore to see if it matches the reference file that was uploaded. If it's a match, it will continue sequencing that strand and move on to the next one. If it doesn't match, the strand will be ejected from that pore and freed up for another strand. You can either choose to enrich for your reference sequence, or you can deplete it, which becomes very useful for projects where you might have a lot of host contamination and you're looking for a way to deplete the host genome without having to incorporate that into your extraction process, such as human gut microbiome sequencing, for example. Data analysis, there are three main options: cloud-based Epitome, Epitome Labs, which is a local version, and open source. Epitome is cloud-based and provides real-time or post-run pre-configured workflows and returns interactive, simplified, shareable reports. You would only need to install the Epitome agent for data upload and starting the runs. Now, Epitome Labs is local and can be installed on your laptop, sequencing device, cluster, or cloud. It mainly has post-run workflows, but has started to provide real-time ones too. Workflows are a bit more configurable than cloud-based Epitome, and outputs such as reports and output files are a bit more detailed. And for open source, there's GitHub. So GitHub tools sit in our nanopore repository and provide the latest pipelines, base callers, modified base detection tools, and many other research resources. These may require bioinformatics expertise for implementation in user-defined workflows. Here's some of our headline accuracy numbers for different research areas. You can achieve high accuracy throughout all investigation types. For instance, we're achieving over 99.9% SNV F1 scores. Our human genome assembly is reaching close to Q50. Methylation detection is over 99.5% accurate, more accurate than bisulfite sequencing. And our microbial assembly has now reached over Q50 accuracy. And the best part is that all this can be achieved by our new chemistry. Also happy to announce that we've partnered with Avantor for the distribution of our MinION and Flongle devices, along with associated consumables. These products can now be purchased directly through their website at vwr.com. And if you have any questions about the products, please feel free to contact your local Avantor rep or reach out to me and I'll be happy to connect you with the appropriate contacts to get you started. I'll now give the floor to our guest speaker, Dr. Lei Liu, to present on his research and how our new chemistry upgrades have been able to support his projects.
Hi everyone, it's my great pleasure to be here today and share my experience with nanopore sequencing. My name is Lei. Currently, I'm a postdoc in Center for Microbial Community at Aalborg University. The Environmental Microbiome Engineering and Biotechnology Laboratory at the University of Hong Kong is the lab where I am from, and most research contents in this webinar that I'm going to share today are from here. In this presentation, I'm going to focus on the topic of deciphering environmental microbiomes using nanopore sequencing, specifically highlighting the complete and high-quality genome reconstruction. Here are the main contents, including self-introduction and introduction to genome-centric metagenomics, followed by how to use nanopore sequencing and take-home messages. I completed my PhD degree at Hong Kong U in 2021 and continued as a postdoc in the same lab for another 1.5 years. During my research, I primarily focused on genome-centric metagenomics. Initially, I tried to use parallel sequencing to recover genomes from environmental samples, but at that time, I found it's too expensive to afford, so I switched to a hybrid strategy, leveraging the Illumina short reads and the April nanopore long reads. Then I continued to work on nanopore sequencing-based complete metagenomics. Since last November, I have joined AAU and been involved in Metas Global and Rethink projects. For this nanopore sequencing-based complete metagenomics, on the one hand, at the microbial community level, we hope to recover at least 90% of the microbial community from the studied samples. For this purpose, we firstly established the feasibility of using only nanopore long reads to recover high-quality genome from environmental samples. I will share these contents later. We also tried ultra-long DNA extraction for better assemblies and nanopore adaptive sampling for enriching the low abundance microbes. On the other hand, at the microbial cell level, we are interested in not only the chromosome, but also the actual chromosomal elements such as plasmid and bacteriophage by trying the POSIS strategy. When sequencing meets the environmental samples, genome-centric metagenomics has drawn more and more attention. Let's first see what's the metagenomics. Metagenomics is the study of all genetic materials recovered directly from environmental samples, and it can be used to answer two important questions: who are they, and what are they potentially doing? As we all know, the majority of the microbes cannot be cultured. At least at this time point, they cannot be cultured in an easy way. Metagenomics, as a culture-independent approach, is a powerful lens allowing us to access the microbial dark matter, the uncultivated majority. Due to the rapidly decreased sequencing cost and the robust analysis pipeline, metagenomics has been cost-effective and time-efficient. The word metagenome or some of the genome, it's the genome derived from environmental samples. Besides genome-centric metagenomics, gene-centric or unbind metagenomics is also very popular in this field, but for this method, it's really difficult to assign genes to a certain microorganisms, and it cannot provide an overall context. Here is a typical workflow for gene-centric metagenomics. Based on the study design, we collect the sample and extract the DNA and perform the DNA sequencing. After QC, we can get the clean reads. These clean reads will be assembled into contig, followed by binning and quality assessment. After we got the max, we can do the following analysis, such as phylogeny and metabolic potential profiling at the genome level. We can see the genome reconstruction is very important in this process and greatly influences downstream analysis. However, there are a few limitations of the traditional short-read sequencing, like the mags are always highly fragmented, and the situation got worse in complex environmental samples. We can see from this figure. One mag normally contains 100 contigs, and sometimes even more than 1,000 contigs. The genome gaps between these contigs indicated the loss of genetic information. Even sometimes we had the opportunity to recover a complete genome using the traditional short-read sequencing. It requires intensive polishing and manual curation, which is difficult to apply for the large-scale studies. It's also very difficult to recover real high-quality genomes. We can see here, according to the MIMAC standard, a high-quality MAC should meet the following criteria. The MAC should be at least 90% of completeness and less than 5% contamination. Meanwhile, at least 18 tRNA genes should be included, as well as the presence of 5S, 16S, and 20S RNA genes. In most studies, only the first two criteria were considered. As I mentioned, it's not an easy task to recover real high quality. We can see from this Nature paper, the authors recovered nearly 100,000 marks, while only less than 0.3 could be classified as high quality, mostly due to the assembly and binding issues when using the traditional short-read sequencing method. The good news is with the technological advancement, some long-read sequencing providers, including PacBio and nanopore, started to pop up. Although they were founded in 2004 and 2005, launching their first commercial sequencing platforms to quite a long time. Here's a summary of the early sequencing products in my former lab at Hong Kong U. As the name suggests, long-read sequencing has a great advantage in sequencing length. However, they both suffered from the sequencing yield at the early stage. We can see here and here less than 1 megabase per sequencing cell. So it's difficult to directly use them for genome reconstruction from environmental samples. Additionally, for metagenomic studies, we also care about sequencing accuracy. PacBio is very famous for its sequencing accuracy, generating long reads with accuracy of more than Q20, comparable with Illumina sequencing. For a long time, nanopore platforms was plagued by the sequencing accuracy, but caught up very quickly in the throughput. Here is a study published in Nature Biotechnology. The author uses PacBio sequencing to generate the high-quality mags from this sheep fecal sample. In total, 54 PacBio smart cells were used, which means a lot of sequencing cost. I don't think it is affordable for most research labs. In contrast, the low entry and the sequencing cost facilitates nanopore sequencing accessible for most research labs. We focus more on the nanopore sequencing, not only because we have seen the increasing sequencing throughput, but also having nanopore sequencing platforms in the lab means that we don't need to wait a few months for sequencing data released from the commercial company. It's almost real time. We sequence the DNA and get the data in 2 or 3 days. After that, we can start to analyze our data. So the question becomes how we can apply nanopore sequencing in the study to decipher the microbial community. Our first try is the genome reconstruction from the PNA bioreactor and enrichment system. This system is used for nitrogen removal from wastewater. We deeply sequenced the PNA sample in 2018, including more than 110 gigabase Illumina short reads and nearly 80 gigabase nanopore long reads. Definitely, we could recover mags using only short reads, by using only long reads did not work due to the, we can see here, the high sequencing error rate. So at that time, we think the hybrid strategy, leveraging the air-prone nanopore long reads and the Illumina high accuracy short reads is deserved to try and may serve as a temporary solution for genome reconstruction. In this study, 13 single contig mags has been reconstructed from the PNA bioreactor. Among them, 8 were circular metagenomes. This is one of the exciting results of this research work because it's really challenging to reconstruct complete genome directly from metagenomic datasets before. Now this hybrid approach enables the high accuracy complete genome reconstruction from this enrichment system. This picture illustrated the first complete genome of the genus Candidatus broccadia. We can see 13 identical transposes with a length of more than 2100 base pair were identified on this complete genome. This may explain why only highly fragmented mags had been recovered for this genus previously. Also, this demonstrated the promise of using nanopore long reads to resolve the long repeat regions. We also try to use the hybrid strategy to resolve the complex activity sludge sample. Quite different from PNA bioreactor, activity sludge harbors thousands of microbial species representing a very highly complex system. We also had a very deeply sequenced dataset for its AS sample. We can see here. Targeting such high complexity, a new hybrid assembly framework was designed. The key idea of this approach was to assign sequencing risks to bacteria candidates. In this way, a metagenome assembly has been divided into many miniature assembly process of bacterial isolates. This method also reduced the requirement of computing resources. Using this approach, 557 mags, representing more than 60% of the microbial community, has been reconstructed, including 14 circular genomes and more than 100 high-quality genomes, encoding the full-length RNA gene operons. We also noticed that full-length CTSRN genes were identified in more than 400 mags, providing the potential to link the CTS applicant to its whole genome for the future studies. Based on the recovered mags, biosynthetic potential of the AS microbial community has also been investigated. In this study, we can see here the length of identified BGCs, the biosynthetic gene cluster, ranged from 10 to 50 kilobase pair. This length would largely increase the BGC detection difficulty from the traditional methods. Specifically, we noticed this kind of phyla encoded a high diversity of BGCs and had a high encoding density, indicating this kind of microorganisms is a high priority for future investigation, taking up the natural products. The primary concern in the application of nanopore sequencing earlier is the high sequencing error rate. As I mentioned before, the median sequencing accuracy was below 90% in 2019. So in my first two cases, we used the hybrid strategy. The short read sequencing should be included. However, based on the sequencing dataset of the MAL community in 2021, we have seen the median value of the sequencing accuracy has been substantially improved, mostly due to the base color upgrades and the newly developed chemistry, Q20 plus chemistry. We can see the high accuracy of the long-reads nanopore sequencing, especially for K9 chemistry. The accuracy has been improved to nearly 96%, and for Q20 plus chemistry, we can see the accuracy has been improved to 98%. The high accuracy of nanopore sequencing makes it possible to use only nanopore long reads to resolve the high-quality genomes from environmental samples. And here is the sequencing summary for our dataset. The first one is the MAL community, including 21 different microbial strains, and the second one is the activity slide sample. To facilitate the rapid genome reconstruction, we introduced NanoPhase, a package to generate mags from nanopore long-read datasets. We first tested the performance of NanoPhase using the MAL community. The results showed that NanoPhase can recover near-perfect genomes. The median, we can see here, the median genome completeness of both mags from K9 and Q20 plus datasets were 99%. We also see the index has been decreased significantly, especially when we use the Q20 plus chemistry. For the complex AS metagenome, 275 mags were reconstructed, and with the median genome completeness of 90%. The median N50 of these recovered mags has more than 700 kilobase pair, which means tens of fold improvement compared to the traditional short-read-based mags. Remarkably, we also noticed that the median counting number in each mag was 9, indicating long reads greatly close the genome gaps and help to recover the near-complete genomes. More importantly, we also observed that the short-read polishing only improved the genome quality slightly, which means in the future, short reads were not necessary to be included in the metagenomic studies. NanoPhase can be easily installed via Conda or Mamba. I would strongly suggest use Mamba because it's much faster than Conda. Here the full model of NanoPhase, including the check, med, isolate model. There are also 2 test models. For this check model, it will check all required dependencies to make sure all the dependencies has been installed before a successful run. For the meta module, it was used for assembly, binning, and genome classification for metagenomic datasets, and the assemblies module was used for bacterial assemblies. Although it was mainly developed for using nanopore long-read only method, we also integrated the hybrid strategy into this package and simplified the process due to the increasing sequencing accuracy. The data shown before has been sequenced more than 1 year ago, so what is the current state of nanopore sequencing based on my sequencing in the past few months? We have seen using the latest sequencing kit, Kit 14, and flow cell R10.4.1 with the GAPI super accurate mode. The median accuracy of nanopore platforms has been improved to more than 98%, and the module accuracy is very close to Q20, 99%. For duplex accuracy, although we did not test it a lot, but based on the data we had, the accuracy of the duplex has been achieved to around Q30. That is quite amazing. We also see the yield of nanopore platforms has been improved a lot. Now for our samples, the median yield for one ProMIS flow cell is around 140 gigabits, which means with the same cost, we can get more high-quality mags. Based on our estimation, the cost per one high-quality mag has been decreased to less than $5. For Minia and the Gradient flow cell, we have seen more than 30 gigabits of sequence can be generated from one flow cell. Here, the last part, take-home messages. Overall, nanopore sequencing is really promising in genome reconstruction and benefit a lot from the improved throughput and accuracy, as well as the theoretically unlimited sequencing length. The pipeline nanophase enables complete and high-quality genome reconstruction from diverse environmental samples, and it supports long-read only and hybrid strategies. Nanopore long-read sequencing, nanopore long-read only methods has been become cost-effective even compared to the traditional widely used Illumina sequencing. We have seen the cost per high-quality mags has already much cheaper than traditional methods. Specifically for genome reconstruction, nanopore-based mags are high contiguity tensorfold improvement in terms of unfifty, and these mags further improved their representation in the microbial community. And taken together, nanopore sequencing prowess could be used to prowess a near-complete genomic blueprint of the studied microbial community. That's the end of my presentation, and thanks for your attention.
Thank you, Stephanie and Lei, for this very interesting presentation. We will proceed now with the Q&A session. Unfortunately, Lei Liu cannot stay for the Q&A session, and questions for him that we cannot answer here will be answered directly by email. So please enter your questions in the chat you have on your control panel. And Stephanie, please, you can start with coming questions.
Yeah, thanks, Maria, and thanks, everybody. Yeah, so we do have just a couple of questions so far. Is it possible to directly detect methylation in metagenomic assemblies? The short answer to that is yes, as long as you avoid PCR, you will maintain methylation information. So our ligation and native barcoding kits are PCR free, and a methylated base will produce a different raw signal from a canonical base. So we're able to capture that information in the FAST5 files. The base modification analysis tool we use is called Remora, and right now it's trained to detect 5MC and 5HMC on DNA samples. So if you're looking for any other modifications outside of those two, like 6MA, there are other third-party software tools you can use, or you can train your own model. And then we have another question. How do you address the inaccuracy of base calling homopolymers? Yeah, so that's a really great question and is actually addressed by our new R10.4.1 nanopores. So those pores have a dual reader head. So we've increased the sensing region within the pore itself. So it's almost like your fragment that's being translocated is being read twice. And so far with our internal studies, we've shown that we can accurately call and keep like Q20 accuracy for homopolymer stretches up to around 7 or 8. So, I mean, homopolymers have notoriously been I would say a challenge in the entire sequencing world, but our new R10.4.1 pore is has been addressing that pretty accurately. Do the nanopores get damaged when using the device? So they are biological proteins. So just naturally, they will degrade as you're sequencing. So when you actually load the flow cell, you are priming the flow cell first before you load your actual sample. And so in that priming mix, there's ATP, there's some electrolytes that you are loading onto the flow cell. Eventually, those will deplete as you're sequencing, and the pores will become dead, so to speak. So over time, the operational life of the flow cell is up to 72 hours. So as you're sequencing, you will naturally deplete the pores on the flow cell. A question about duplex reads. How do I generate duplex reads? Is it a different protocol or chemistry? So duplex capabilities are already built into our new version 14 or V14 kits and the new R10.4.1 pore chemistry. You just need to turn on duplex-based calling in Minnow, which is our software, before starting your sequencing run. This is because we use a different algorithm to produce duplex data and pair up the template and complement strands. Other than turning it on, you don't need to do anything special with your sample. It's all built into the new chemistry. Do the flongles have the new pore chemistry? Yes, they do. Yeah, so flongles right now are available in the R9 and the new R10 version. And we do plan on discontinuing the R9 version by the end of this year, so that R10 will be our new gold standard flow cell. Have you used directly from fecal samples instead of mock communities? Yeah, so I'm not sure if that question was directly pointed towards Lei or if Nanopore has done studies on fecal samples, but we have. And we do have some recommendations for extraction methods as extraction protocols for fecal samples that can actually be found on our community. So if you go to our website, you should be able to log on to the community if you have an account and find that information there, or we can follow up in an email. How many stool samples can be analyzed using one device? It's going to really depend on if you're using, the stool samples for metagenomics. Then it's going to depend on a lot of different factors. I want to say for metagenomic sequencing, a lot of people will multiplex up to either 12 or 24 samples on a single flow cell and still get sufficient coverage. But it is going to be dependent on the sample composition, like if more bacterial or like fungal species are more abundant within your sample, etc. Because of course, the lower abundant species, you'll need to sequence to a higher depth in order to identify those. But for the most part, I see a lot of people multiplex up to 12 or 24 samples. Are there alternative tools to Nanophase, and how do they compare in accuracy and completeness? I will take that question back to Lei and also to our technical support team to see if they have any insight to that as well. So not able to answer that question right now, but we'll definitely get back to you on that. Well, switching to the V14 kit and our 10.4.1 flow cells require us to change the library prep protocol in any way. So for the most part, the library prep has remained the same. We just made some updates to like the sequencing adapter. So now the sequencing adapter in the new V14 kits is what we call a high capture adapter. So it's able to bind and ligate to more of your fragments, which has led to higher outputs from the flow cell using those kits. So overall, the library preparation process should really remain the same. We've just changed the adapter, and I believe some of the input requirements and loading amounts might be a little different, but other than that, I want to say it's about 90% similar to the previous chemistry. See, what method would you recommend for sequencing 7,000 base pair viruses? Would it be possible with nanopore minion? Yeah, absolutely. Yeah, it's such a small genome that it can absolutely be done on the minion or even the flongle flow cells. So the flongle flow cells are just like our minion flow cells, but they contain less nanopores, so they provide less output. So on a flongle, you can probably receive around 1 to 2 total, like gigabases in total from it. So if you're working with a small genome like that, you can probably get that done on the flongle at a more cost-effective way. So how do you eliminate host DNA? So there's a couple of different protocols for that. So there was one protocol that was developed by one of our customers to deplete host DNA when they were looking at upper respiratory viruses. So that's like an upstream host elimination. Another way you can do that is with adaptive sampling. So if you wanted to, let's just say you were working on a human genome and you wanted to deplete the human host DNA within your sample, you can actually upload the human reference genome. So you would prepare your sample as normal as if you were going to do whole genome sequencing, for example, and upload the human reference genome, turn on adaptive sampling, and choose deplete. So if it detects any reads that are translocating through the pore that have the human reference sequence, then it will reverse polarity and eject those strands from the pore and sequence everything else. So you can either choose to do that upstream, like host elimination upstream using a certain extraction process, or you can do it the adaptive sampling way. Are you looking to update the 16S kit to the new technology? Yeah, so we are right now developing, I would say we're exploring the fate of the 16S kit, whether we want to update that to the new chemistry or have a different offering. But in general, there will be a V14 version to do 16S sequencing. What that looks like, we're still trying to figure that out, but we'll definitely keep everybody updated. So do you have any advice for DNA extraction for use with the rapid barcoding kit? I've tried several different methods, but the most successful, I'm getting a few thousand sequences, but these are all short with average read lengths of 1,800 base pairs, where the estimate from the agarose gel are 20 kb. So with the rapid sequencing kit, so it utilizes a transposase, so it will cleave your fragments. So if you're starting with 20 kb fragments, when you use that kit, the transposase complex will simultaneously cleave and attach the sequencing adapters. So there is fragmentation involved in that kit. So that's probably why you're seeing around 1,800 base pairs at the end of the sequencing run. So on average, you can probably expect around like a read length distribution of somewhere between like 1,500 to 8 kb. So that seems about right for that. But it also just depends on also what your starting material is, like the type of sample that you're working with. If you wanted to maintain those 20 kb long reads, then the ligation kit would probably work better. So do you have a recommendation with eliminating rat DNA? I'm not sure if I've ever come across that specifically, but we can dig into that a little bit more. So we can send you the human host depletion DNA protocol that our customer has done, and maybe that can also be tailored and used towards rat DNA. Yes, there will be a CRISPR-Cas9 v14 kit. Okay, I think that was all the questions that we had coming in. Maria, I'm not sure if you wanted to close the session or if you had any closing remarks.
Yes, thank you, Stephanie. I was waiting if there were some more questions coming. If it is not the case, now we will close the session here. And of course, I would like to thank you all, participants and speakers, for your interest and collaboration in this webinar, and hope you have enjoyed and found this webinar useful. So please do not hesitate to contact your Avantor Life Science specialist if you need more information and support about this topic. Thanks and have a nice day.